Well, I'm very happy to be invited to participate in this AI meeting in medicine. It's a very exciting development. I'm going to talk about my own experience in using AI in bioinformatics, structural biology, and drug discovery. My own work focuses on the basic science, really, in my academic lab, underpinning cancer, microbacterial diseases like tuberculosis and leprosy, and more recently in COVID-19. So I'm going to tell you what we've done mainly in the area of AI, which focuses on looking at protein-protein interaction sites and ligand binding and impacts of mutations. So, of course, the background is that we have to have a knowledge of the genomes, and I've worked considerably on moving the genome information into proteome information, which is three-dimensional and can provide the basis of drug discovery. And traditionally, I've been involved in structure-guided methods, particularly ones involving what we call fragment-based discovery in a company that I co-founded now 22 years ago. That's called Aztecs, and we have 2 cancer drugs on the market. But I'm not going to talk about that. I'm going to talk about our work on AI. And this has really 3 areas. One of them is using deep learning methods to look at protein-protein interaction sites. This is quite important and really an exciting area to look at in targeting in drug discovery. Then, of course, ligand binding, and that is a more obvious area. We're looking at small ligands involved in drug discovery. What I should have said, of course, is that the AI deep learning is being led by Aryan Jamasp in my group, and the small molecule binding to sites by Liviu Copiu. And this is really work that's been going over the last couple of years. But for many years, since actually around 2008, but intensely from 2013, then I've been working using AI methods to look at understanding the emergence of resistance. And this is hugely important if you're going to design drugs, and especially in cancer and in antimicrobial drugs, then you've got this problem arising and making the drugs you have ineffective. And one of the methods that we've devised, called MCSM, is through Douglas Pires with David Asher involved, which we started around 8 years ago, and have a whole suite of software. But let me first, in this Center for AI in Medicine, just remind you that the major breakthrough on 2020 on looking at proteins is AlphaFold, and we've all been impressed by the paper in Nature, which uses neural network trains for predictions of distances between pairs of residues, and then that conveys more information than in contact predictions. It has all sorts of interesting aspects that describe the shape of the proteins, and it's optimized by simple gradient descent algorithms to generate structures. And of course, this software really hit the headlines when it won the Critical Assessment of Protein Structure Predictions, CAS 13, which is an annual competition which I've been involved in over the years, but not in this one. And this obviously gives us insights into cases that we wish to design drugs for, but for which we have no structures. So that's the breakthrough there. Our contributions have really been in these specific areas. So the first one I want to mention is the work of Aryan Jamas, a very, very talented PhD student, only in his, I think, still second year. And he's developed some software for a number of applications, protein, ligand, protein. But I want to just discuss the protein-protein interaction work, which we've written up. Of course, protein-protein interactions are very important to biological function. People forget that most regulatory systems in the cell have multiple proteins involved, and that's in order to gain selectivity. And we can target the protein interactions in drug discovery that are critical for those. This is involved in cellular processes, and it gives you very highly sensitive, highly selective drug targets. And we can predict the residues involved in these protein interactions. And this is what Aryan has done. And here's the software on the right-hand side, Scheme, for the deep learning. You have an overview of the approaches for PPI prediction. And you can input the sequence in order to feature the various aspects of the architecture of the model for drug discovery. So that's one area. A second person in my laboratory, Liviu Copiu, has been looking at assessing affinities of ligands. And this has really brought into mind a wide range of applications in life sciences and obviously in drug discovery. But if you look at many current machine learning approaches, they're limited by requiring co-crystals and also outputting a class really binding or no binding, so rather simple. But there are major problems, and I think that what Liviu has done, which is really important, is to look very carefully at the kind of data that people have used. And it's really quite heterogeneous, reporting affinities, defining biophysical values, and really merging different kinds of values. And also quite a lot of it is not very well produced. So a lot of careful curation is required. And that is very critical. There are a number of aspects, and what he's done is to use just the interacting chain. And then he's developed a number of approaches, semi-manual approaches, but also an algorithm that takes into account the 2D contact map between alpha carbons, beta carbons, and side chains. It deals with the protein sequence, which is embedded using a transformer algorithm and trained to predict structure and function. And then the ligand structure is introduced in Smile's format. So these are very important aspects. And I think what we've learned mostly is how careful one has to curate one's learning data to get very useful aspects of assessing the affinities if you're going to use AI. So that's the second area. The third area, of course, has been in my own company, which is the fragment-based discovery in Aztecs from 1999, where we've had from 1 or 2 people up to now over 130 employees, with a whole range of different aspects and 2 drugs on the market for cancer, but where we've begun AI work. And this has convinced me that it is going to be very useful for drug discovery, and these kind of binding assays that I've described are going to be critical. But the area where I've spent most time working is in understanding mutations. We did the first work, as I said, of this about 14 years ago or so. But now, since 2013, Douglas Pires, who came to my lab from Brazil with a computer science background and had an interest in machine learning, has developed software which is called MCSM. There's a huge number of flavors of this. We look at stability, protein-protein interactions, protein nucleic acid, and protein ligand, and look at the impact of mutations on those different features, which have really not been done in quite this way before. And so this was developed initially 7 years ago. This is the one for protein ligands, and it needs a good database to learn from. And that's been the major contribution of David Asher. Both David and Douglas left my lab a few years ago and now are professors in Australia, in Melbourne, and have carried on this work on MCSM, which was originally developed in Cambridge. So we can use these methods to look at the impact of a mutation allosterically, so distant from the binding site, on a protein ligand. And this is an example of some mutations at a protein interface, which are characterized by these red residues. And you can see they can impact on a ligand in a dimeric structure in this case, which is quite distant. So we have many examples of this kind, and this has been a very good example of the use of AI. And we have this software which can look at all these different aspects of mutation impacts, stability of the protein, protein-protein interactions, protein nucleic acid, and very importantly, protein ligand. So that's where we've been on this drug discovery area of using machine learning. It's really produced a very large number of papers, not only in developing the method, but in applying it. And I'm very pleased to see that it impacts on the subject rather well. A lot of my statistical methods we developed earlier on, you can see the machine learning is extremely competitive. So thank you very much for listening to me. I'll stop the presentation now and thank Andres Mejia very much for the contributions they've made in organizing this meeting. Thank you very much. Also to Sean O'Neill, who's helped me get the presentation set up. Thank you very much.